Hello everyone, welcome to yet another new episode of Genetics and Evolution. So today's topic will be chromosomal theory of inheritance. It's a very important topic, so please stay tuned throughout the full episode because it is going to be a very important and very interesting episode. So what we are going to learn today, we are going to learn about the principles of inheritance, what we have already spoken. We will just summarize the principles, so summarization of the basic principles of Mendel, and we will see the work of Hugo DeVries, Correns and VonTschermak, and we will see why these Mendel's theory were unacceptable by many of the biologists and why they couldn't accept the principles of Mendel's law. So we will see each and every law and what were their flaws. So 3 laws we have already studied in Mendel's inheritance part, and then we will finally come to the final part, that is chromosomal theory of Walter Sutton and Theodore Boveri. So this is the main part of today's lesson. So before that we need to summarize these 2 parts as well. So don't skip in between and stay tuned throughout the full episode, and please don't forget to subscribe to my channel so that you'll be getting many more interesting topics like this. So let's get started. So this is Gregor Johann Mendel. You have studied already in the previous episodes what he has done, his monohybrid cross, his dihybrid cross and all. We have studied about the Punnett square. So if you have not yet gone through those episodes, I'll be putting it in the description box below as well as in the I-link above. So here he has published his work on inheritance of characters in 1865. But till 1900 it was unrecognized. So what was the cause of this work not being recognized? One of the thing is that then in 1900s the mode of communication was not easy. So as it is now, it is very easy. When you are doing any work, you can publish it online. So it was not publicized at that time. Secondly, his concepts of gene, what he told was factors. So Mendel has told factors or traits. Okay, so he told that these genes or factors, they have as stable and discrete units and that control the expression of the traits and of the pair of alleles which do not blend with each other was not accepted. So he told that these gene or factors, they are not blending into each other. They are carried as it is from the parental generation to the F1 generation and from that to the F2 generation. So this main part was not accepted by the contemporaries. So and as the explanation for apparently continuous variation was seen in nature. So as continuous variation was seen in nature, that from generation to generation there was a variation. So if these factors or genes, which are now called genes, was same, were stable, they were not having any changes, or if they were not blending, so how can these variation in nature happen? What we can see today. So this main part was not accepted by his the other contemporary scientists. Okay. Another thing was that Mendel has used mathematics to explain biological phenomena. So another thing was that he used mathematics. Okay, so he used mathematics to explain the biological formula. So biological things were explained in mathematics. So this approach again was not at all acceptable by the biologists of that time, of his time. So finally Mendel's work, it got suggested that these factors or genes, he could not provide any physical proof for the existence of the factors. So it was lacking the physical proof. So lack of physical proof was there. Okay, so lack of physical proof was there for his so-called factors, which are now called genes. So he couldn't prove that. So then what happened? So in 1865 it was done. So then again in 1900 what happened? We will see that. So first let us do a quick revision of these 3 laws. So first one was the law of dominance, second one was the law of segregation, Then after the dihybrid cross, after taking two pairs of traits, Mendel has proposed the law of independent assortment. So just we will do a quick revision of that, then we will move on to the other scientists' work from which we have derived the chromosomal law of inheritance. Okay. So first here comes the law of dominance. So we have already told that characters are controlled by discrete unit called factors. So these discrete units are called factors, and factors occur in pairs. You have seen that all the factors are occurring in pairs. Now in a dissimilar pair of factor, one member of the pair dominates the recessive. So here we can see that both the pairs are similar. So both are written as capital A. So both are dominant. So there is no question of dominating the other one. So it's a pair having both the dominant alleles as well. So the phenotype will be dominant only. And genotype is this. Here both are recessive. Both are written in the lower case. Okay. So they are showing this green apple. Okay. And the phenotype is this and the genotype is this. Now here you can see the two alleles which are of different type. So it is, we can say that one is dominant and another one is recessive, right? So here the dissimilar pair of factors are there. So what we were talking about, and this dissimilar pair of factors where we can see the A, which is in the dominant case, which is dominating. So the color of this is red. So the phenotype or the external feature will be red, whereas the genotype is having two characters. This A is recessive. A is dominating over the other. So that is the law of dominance. Now after this he told the law of segregation. So what was that? So this is the law of segregation. Segregation, what is happening? Now though the parents they are containing two alleles, during meiosis, the factors of the alleles, the pairs are separating and they are segregating from each other and others, such that each gamete is receiving only one of the two factors. Each gamete is receiving one of the two factors. Okay, so of course a homozygous parent produces all gametes that are similar, while a heterozygous will produce two kinds of gametes. So if it is a homozygous, it would have produced only one kind of gametes, but as they are heterozygous, we can see that two kinds of gametes are produced and having one allele with equal proportion. So one allele is present there in equal proportion. That is, the two copies of each genetic factor, they are segregating during the development of the gamete. So they are separating during the formation of a gamete. Okay. And to ensure that each parent's offspring, they will attain one factor each. Okay, they are segregating. They are ensuring that each parent's they will be getting the one factor from each parent. Okay, they are segregating. So they'll get the maternal alleles as well as the paternal alleles. They are segregating. They are separating equally. So if that is a heterozygous parent, they will have two gametes, two kinds of gametes. If it is a homozygous, they will have all kinds of similar gametes. Is it clear? So this is the law of segregation. Then we will come to the law of independent assortment. So here we are with the third law which he proposed after the dihybrid cross. That is after taking two pairs or two traits of characters. So this is the law of independent assortment, right? So based upon the dihybrid cross, Mendel, what he proposed, that when two pairs of traits, here we can see that two pairs of traits are being taken. One is capital R small r, one is capital Y small y. So this is, we can say that round, capital 1 is round and small r is wrinkled. Capital Y is yellow and small y is green. So this we have already studied in the previous class. So just we will do a quick revision. And if you have not gone through the previous class, please do go through it. I'll be putting the link here down in the description box. And here that law of independent assortment has been done in quite detail. So here we are just going through a quick recap. So what he told, he refers to the event in meiosis where the alleles assort independently. So this is meiosis is happening. So RRYY, this is the female part and this is the male gamete. So these all are having they are after meiosis. We can say 50% of the gametes have R, 50% are having small r, 50% are having Y, and 50% are having capital Y. So this is what is having. So we all know that meiosis is a reductional division, but at the end of this meiosis we can see that all this gametes, they are having all the parts in 50%. Okay, so besides each gamete, either R or R, you can say that Y or Y, they are having independently. So R is independent of this R and this Y is independent of this Y. So therefore we can say that 50% of R bearing gamete has Y. So 50% of small r bearing gamete has capital Y, 50% of capital R bearing gamete has capital Y, 50% of capital R bearing gamete has small y, okay, and 50% of small r bearing gamete has small y. So this is what has happened, okay. So thus after the final product, what is happening? So the 4 types of gametes are there. So 25% of the total, like each gamete is having 25%. Okay. So that will come to like this: RY, RY, small r, capital Y, and small r, small y. So like this, we can see that this is going to happen, and this has happened. That is, the frequency is like that: 25% or 1/4 of the total gametes has been produced. So this we have already studied in the Punnett square. So just we will read once this, then we will move on to the studies of the Hugo DeVries, Correns and VonTschermak. Okay. So what he has proposed, we have studied about the law of independent assortment, which is stating that alleles of 2 or different genes, that is 2 genes we have seen, alleles of that, they get assorted into gametes independently, right? So they are independent of one another. In other words, the allele of a gamete receives for one gene which does not influence the allele received for another gene. So they are not being influenced by the other. They each and every one is individual. They are having their individuality. They are independent of one another. Okay, so this we have studied relating to the P color and P shape. So that is what I have discussed earlier. So RR and YY. So this is what we have studied. Okay, so round and wrinkled and yellow and green. Okay. So this is what we have studied earlier in the Mendel's law of segregation. So I'll not repeat it. So please go to that episode and read. So let us proceed with the next part. So in 1900, what happened? Rediscovery of Mendel's work was done. So these 3 scientists, 3 botanists were there: Hugo DeVries, Carl Correns and Erich von Tschermak. So they independently rediscovered Mendel's work in the same year. And a generation after Mendel's published his work. So they kept on working after one generation. In 1900, they helped to expand the awareness of Mendelian laws of inheritance in a scientific world. But these 3 Europeans, they were unknown to each other. They were working on different plant hybrids. And when they each worked out on the laws of inheritance, what did they review? They reviewed that the literature before publishing their own results, they were startled to find Mendel's old papers spelling out those laws in detail. So before they published their own results, they saw the old papers of Mendel's, and each man announced that Mendel's discoveries and his own work as a confirmation of them. Then Mendel's work got confirmation. Okay, so by 1900, several cells, chromosomes were sufficiently being understood, and to give Mendel's abstract ideas a physical context. So these Europeans, they, along with their papers, they studied Mendel's old papers as well. They found out that his discoveries were also it was good, and his discoveries were being confirmed by them. And Mendel's abstract ideas, which were abstract, they got a physical context. Okay, so is it clear? Now after this led to the discovery. After this, advancement of microscope was taking place. So then many advanced microscopes came into being. So advanced microscopes were there. And what happened then? People were able to see the cell division under this microscope. Okay, so they used to see the cell division. So cell division was visible under the microscope, and we can see that the structures and the nucleus that appear double, they divide. So just before each and every cell division, this was all clear, and they were called chromosomes. So this all were discovered during this time, and they were visualized by the process of staining. So staining was done. Chromosomes used to take the stain, and this full process was being visible under the microscope. So then after this Walter Sutton and Boveri in the year 1902. So 1902 they came with their chromosomal law of inheritance. So let us move on to that part. So these are the two scientists, Sutton and Boveri, who proposed the chromosomal theory of inheritance. Okay. So what they did, they in 1902, they explained that the behavior of the chromosomes and the genes was similar, and as per the law of Mendel. And if you recall that, you know that mitosis is an equational division, and where meiosis is a reductional division. So the two alleles, the alleles of a gene, the pair are located on a homologous chromosomes. We can see all the homologous chromosomes over there. In this part, Y is located. That is the loci of this chromosome where this gene Y is located. Here again, in this loci of this chromosome, this white part, the small y is located. Like this, R is also located in this specific loci of the chromosome, and small r is also located in the specific loci of this chromosome. So this is how this 4 chromosomes in meiosis 1, they are divided into 2 and 2 each in meiosis 1. This also in meiosis 1, they are divided equally, but they are reductional division, because the 4 they combine, they form 2 each. Again in the next division, meiosis 2, they divide and they are having one strand each. Okay, so there is a reductional division, but they are segregating in which manner? That is 1/4 YR, 1/4 small y small r, 1/4 capital Y small r, and 1/4 small y capital R are there. So they are segregating and they are independently assorting. So as per Mendel's law also, we have taken 2 pairs of contrasting characters, traits. So they are having genes on this specific loci which are independently assorting, and they are moving on into next generation, and during the formation of the germ cells, they are having 1/4 of this separate genes which are independently assorting, right? Is it clear to all of you? Now we will move on as per your NCERT book, which will be easier for you to understand. So just you can recall what we have learned in the previous classes when we have studied about the cell division. We have studied the behavior of the chromosomes during mitosis as equational division, but during meiosis it is reductional division. So the important things to remember that chromosomes as well as genes, they always occur in pairs. You can see here now during multiplication they are duplicating, they are in pairs. Okay, so they are in pairs. Okay, is it clear? So they are all in pairs, and here when they are in the anaphase part, we can see that they are in pairs with all the 4 characters or 4 genes together, which will be divided into each gametes. Okay, so 2 each we have seen here. Again they are segregating 1 1 1 1. So each are segregating with the 25% each. What we have told in the law of segregation, that they are segregating, they are having 25% each. Okay, so 25% each carrying the characters in them, and they are not blending, they are not mixing with each other, and they are all located on the homologous sites on homologous chromosomes. So all are homologous chromosomes. They are not heterogeneous chromosomes. They are all homologous chromosomes, and they are having two alleles of a gene pair. So this is what is happening. This is what the germ cell distributions is having, with all 4 chromosomes are there in this 4 cells. So germ cell formation is being done. Now next, there is a comparison between the behavior of the chromosome and that of the gene. So what is happening here? You can see in A and B, so both the chromosomes occur in pairs, genes also they occur in pairs. Chromosomes, what they are doing, they are segregating at the time of gamete formation, such as only one of the each pair is transmitted to the gamete. So this is what we have seen here. One of the each pair is transmitted to the gamete. Okay, so this is what is in case of the gamete formation. In gene case also, one of each pair is transmitted to the gamete. Independent pairs, they segregate independently of each other. Okay, and here also same thing, one pair segregates independently of the another pair. So this is what we have seen. Quite similarity between the behavior of the chromosomes and genes. Okay, so can you tell which one of these columns A and B represent chromosome and which is representing the gene? How did you decide? So they are not separate. So the gene character and the chromosomes character are almost the same. So what is a gene and what is a chromosome? So that is what these Sutton and Boveri, they told specifically what is gene. So before that, we will see there are 2 possibilities. So possibility 1 and possibility 2. So let us move on to those. So what is happening during the anaphase of meiosis 1? The 2 chromosome pairs can align at a metaphase plate. Okay, so they are independent of each other. They are meiosis 1 in anaphase. They are independent of each other. They are we can say that to understand this, compare the chromosomes of 4 different color in the left and the right column. So in the left column as well as in the right column, they are all separately, independently aligning in the metaphase plate. Okay, so in the left, the colors are, we can say that one long orange, one short green, one long yellow, and one short red chromosome is there. Whereas in the right one, possibility 2, this can also happen, that one long orange and one short red and one long yellow and one short green is there. So this is the possibility. So meiosis 2 in anaphase, what we can see, that we have seen in the cell division, that in the centriole part it has divided, it has segregated separately. So the strands have become separate. Now they have separated from each other. So their binding in the centriole part is not there. Then they are being individually, each and every chromosome individually has been given to the germ cell. So one in each. So orange and green combination of chromosomes, yellow and red, whereas it can happen in possibility 2 as well. Orange and red and yellow and short green can also happen. So this is a possibility 1 and possibility 2 which can happen in the meiosis 1 phase, 2 phase, as well as while giving it to the germ cell. So these are the 2 possibilities which can happen. Okay, so this is how the independent assortment of chromosomes of Mendelian's law was established. Okay, so Now, finally, what we get in this chromosomal theory of inheritance, we get that it states that what are genes? Genes are located at a specific loci on the chromosome. So, if suppose this is a chromosome, on this specific loci of chromosome, we can see the genes like A and A. Okay, so these are present on the specific loci of a chromosome. So we can just draw another one. So this is another chromosome. On that we can see a gene. We can see a gene like this on a specific loci. You can say this is B and this is small b. So this is what was first proposal of this chromosomal theory of inheritance. So it is a foundational concept in genetics. So it was just a foundation proposing that chromosomes are the carriers of genes. So the chromosomes are only carrying the genes from one generation to another, and their behavior during the cell division, it explains the patterns of inheritance. So how the next generation, the F1 generation, the F2 generation and so on will be, the pattern of inheritance was proved by this chromosomal theory of inheritance, and it was observed by Mendel. It was observed, and this theory was primarily developed by Theodore Boveri and Walter Sutton in 20th century that we have studied now, that is in 1902, based on observations of chromosome behavior during meiosis. So we have just now proposed the two meiosis probability in meiosis 1 and meiosis 2 phase, and how they are being given the chromosomes in the germ cells. Okay, so it is the chromosomes that segregate or separate and assort and distribute into the groups of same kind. So like that they were given to the germ cells. They were segregating, they were separating independently during meiosis and recombining at the time of fertilization in the zygote. So in the zygote, in the germ cell, how they were fertilizing, how they were moving on, we have just seen. So this, how they are recombining again, and how these genes are, specific genes are linked to this chromosome, that is linkage, and we can say recombination is the next topic, and it is a very important and interesting topic as well. So this was proved by Morgan and his colleagues when they worked on fruit flies. Okay, so on fruit flies they worked, and hence it was proved by Morgan and his colleagues, and this is the theory of inheritance. Okay, so now this chromosomal theory has been proved, as we have seen, as it was proved by the experiments on Drosophila melanogaster by Thomas Morgan. So in the next class we will be coming to this important topic of linkage and recombination, which is very important for you all, and you should know this in detail. So for that you need to subscribe to my channel, stay tuned, stay updated with all the episodes which I am posting for you all. So till then, keep watching my videos and don't skip in between, because throughout the video there has been many informative topics which have been taught to you. So thanks a lot for watching, and stay tuned, stay subscribed, and keep on watching my videos.